like the moon and the sun. She is worth more than gold, like the stars in the sky.
Good afternoon, colleagues.
Good afternoon.
Yes, I believe everybody here is in drug production and quality assurance. And so this is our class today. And we'll be talking on our guest lecturer today is no other person than Dr. Nonye Onyewuenyi.
Dr. Nonye Onyewuenyi is a strategic and results-oriented pharmaceutical R&D scientist and project management executive with over 25 years of leadership across global drug development, analytical R&D, and cross-functional program execution. He has proven success guiding end-to-end product development from preclinical through commercialization, integrating scientific depth with operational governance. He's an expert in aligning global stakeholders, driving portfolio value realization, and managing complex multi-program pipelines across US, EU, and Asia. He's adept at leading PMO initiatives, optimizing resources, and achieving financial discipline across R&D operations. He is recognized for decisive leadership, analytical acumen, and ability to deliver innovative solutions and accelerate timelines, ensure compliance, and achieve business objectives. Ladies and gentlemen, I present to you Dr. Nonye Onyewuenyi.
Thank you very much. Let's just start rolling. So the slide you're looking at, I just wanted to showcase what we do in these 2 days. The first one will be talking about the principles of drug formulation and design, and then it pressed it upon me to have like a mini workshop on WHO pre-qualification. And the rationale for that is, if you are able to make a product and produce it, I don't see why you cannot be able to market it in other parts of the globe. And being able to do that is to be able to successfully pass or go through the WHO pre-qualification. And that starts with you understanding the drug product you have produced, understanding the process as well as the product. Because once you are going to do your submission with the agency, you must be able to package your module 3, which has the CMC document and all the test, analytical and formulation data packaged effectively for the agency to believe that you understand what you have submitted. So that will be one of the case studies. But within there is about 5 or 6 different case studies from 6 countries in Africa. I think in Nigeria we only have 1, which is personally I think it's very disappointing. But okay, and then for tomorrow we will really go down. We will talk about the QBD, but we will drill down to so many explanations of QBD, and most of you will see that once you start your drug production in the lab, in your manufacturing site, you do not just focus on the formulation, you focus on the analytical techniques or the analytics, because 2 of them, they run parallel to be able to ensure that at the end you understand the process is optimized, and you are working within the design space, and within the analytical, you are working within the operating range of all your analytical tools. And then also for today, we will also look at how can artificial intelligence in drug manufacturing. I know that it's still very new, but it's something that you must embrace. And I will go through the basic fundamentals and also to give you some tools which you already, some of you know, FMEA, right? Failure modes and effects analysis. You understand the Ishikawa diagram. There is basic, DOE. You understand design of experiments. Those simple things will help you have a thorough understanding before you finish your project. I will start by, first of all, as a mother, as a student. I was a student like you guys before. We know that most young children, when they are growing up, they want to know, am I really a scientist? And I know that some people, because of the challenges they have, they may not understand, that they are. So the simple analogy I always use in drug development is, as a mother, and each one of you, not saying that men don't cook, when you are going to make your soup or food in the kitchen, you first of all list out all the ingredients, all the materials. And I was talking to somebody yesterday, if I want to eat the bitter leaf soup from Anambra State, I cannot go and eat the bitter leaf soup from Imo State, thinking that I'll eat the same quality type of soup. They all use bitter leaf and all those things, but to make a bitter leaf soup from Anambra State, they have to have the materials qualified, the right ones, and including the ogiri. What do you call ogiri? I don't know what you call it, but the message I'm trying to relay is that every product you're going to make, you must list them out one by one before you do anything, and then try to characterize those materials. And then after understanding and characterizing each material, you now have to understand the interaction between both the excipients and the drug and excipients. That's when we talk about drug-excipient interaction studies. If you are able to understand the materials, why it is hygroscopic, why there's too much, each and every one of them, then you can eliminate those things that will not be well for your product. So this is the fundamental of where to begin. Are we clear in that? This is the principles of drug formulation and design. So these are the three main areas the organization or the management or whoever. The formulation has to bridge. First of all, let me say, if you have a raw material, the API is not automatically a drug product. And the typical example is the insulin that I put there. If you swallow raw insulin as opposed to the formulated product, you are really creating a problem for yourself. And then the next thing is, however you do your formulation, determines if you will have a good product to be able to give you a good performance at the clinic. And lastly, to have a good formulation now strategically makes it imperative that locally you can have product. You should be able to have product. And so as a scientist or you developing the drug, this symbol means how, and how are you thinking. So the scientist has to think through to define what the formulation they are going to make based on the customer requirements. And what do I mean by customer requirements? Some people cannot take tablets, they cannot take capsule, they can only take liquid or injections. So those are the customer requirements which actually compels what kind of drug product you end up with. So when it says integration of API and excipients into a dosage form, please hear me clearly. And generic organizations where they have made Johnson & Johnson, Novartis have already done the innovator, the brand. And then some people have the misconception that, okay, it has been done before. Let me just mix all the excipients and do my tablet compression. I have a tablet. People that have chemists all over Nigeria, sometimes they do that. So, but the integration means really understanding the mixture of the API and the excipients to have them properly integrated so that you are having one product that is supposed to be the medicine. And then the customer requirements, the type of dosage form depends on what your organization wants, what the patients need. That's why it is supposed to be patient-centric, because some patients only go with IV. Some may not take swallow, they cannot. And then it's only when these two are successfully done that you have, you will ensure there's safety, there's efficacy and stability of the product. Remember, we live in a very tropical climate. So what are the objectives? We want to understand, establish core formulation principles, link science to quality outcomes, and finally to align whatever we are doing to meet regulatory expectations. If after all these things you develop a product and you do your submission with the agency, they will look at it, they say all these things is rubbish because you did not align with their expectation. Why drug formulation is Nigeria's most critical investment? Very critical. It's the last speaker, we can compete, but if we do not start it from here, the understanding, we are not going to go anywhere. So, as we all know that the Nigerian pharmaceutical landscape, over 70% of the drugs are imported, and they are coming from India. And the money that is spent, it's more than too much. Just importing drugs, they are in there, like in where I work, you make a call to the call center, it's answered in India or Philippines. So they are outsourcing everything, and we are busy coughing out the money. So the opportunity for us is there, because we have the excipients, we have like the cassava and all those, we have the raw materials, but how do we harness it? How do we now go into production? So what is drug formulation? Without looking at that, can you easily say what it is? It says there is an art, science and art, but it's not only science and art, there's also engineering. Why did I say engineering? When you are developing control release formulations, right, it's actually engineering. I'm going to use one of the examples that I worked on, Concerta, the one that has Oros technology, that where you have to drill a hole or use a polymer that is pH sensitive, that absorbs water and pushes out your drug. All those are engineered. So it's not just the art, but to understand the science. So the drug formulation is simply using these two things, including your engineering, to form a medicine that is safe, effective, stable, and good to the customer requirement, to the patient. So, and under these three, you see, it has to be safe. So your excipient understanding, there's no toxic degradants. It is a stable formulation, and most importantly, has to be compatible. And one of the things of QBD is, yes, there can be many excipients that have the same properties, but you have to evaluate them, evaluate the interaction, and pick the best one that gives you the right number. That's why there has to be compatible excipients. And then under efficacy, is it bioavailable? If you are doing the generic, you can avoid, just they have done the bioavailability, the BE studies. You don't have to, once you have that information, because if your product is not bioavailable, you're just wasting time. So it has to be, the release profile has to be maximally, if you want it to release, if it's a controlled one, you want it to release 20% in 30 minutes, 40% or 70% in half time. Just everything has to be controlled. And if you want it to go straight to the heart or straight to the stomach or wherever, the large intestine or the small intestine, it has to be targeted. If there is a cancer cell, you want that drug to be engineered to target that. And then, is it manufactured? And that means, did you understand your process so that you can, once you do it in the pilot plant or on your bench scale, can you move it now to manufacturing? And what if you push all your processes and you understand them, then scale it up to manufacturing? does it produce and behave the same way as what you did on the bench? If you do not have easy manufacturability or transferability, you have not started. You are going back to square one and you spend money, wasted money, trying to do the formulation. So make sure that it's thoroughly evaluated at the onset. And then, is it compliant? Okay. Cost effective. If you mess up, and your company will probably fire you if you do not do the right thing. I don't know how they fire here, but over there they'll just call you at the conference room and tell you, tell you all sorts of rubbish. Okay. So why does it matter? So it matters because of everything that I've said. The drug we are going to make from the API, you are just, the formulation is a bridge to the real thing. To make it the product that the patient can use. Is it manufactured in such a way that it's safe, its efficacy, and you can, when a patient starts using it, can they comply? Can they, because you want patients to comply to the use of the drug or whatever is prescribed. And then, how can you compete? Can you submit your dossier to NAFDAC? And like I said, globally, then you can submit to United States or submit to Singapore, South Korea, Japan, for instance. So, can you be competitive? Okay, push it. So, there are two dimensions in drug formulation. If you are able to develop your product, can it perform all those attributes? Let's say the paracetamol. Does it dissolve and give you that release profile that's expected? Solubility and dissolution, it governs how quickly drug becomes available. If I take the Tylenol or the Panadol, which is very simple, it's highly soluble, we are going to look at the BCS classification. If you take it, does it dissolve and then it starts going to the right places? And then, can it be developed? Which means you have to understand the physical and chemical properties and stability of the API. Then, if you do that, can you easily transfer it to the contract manufacturing organization, that is from your simple lab bench to scale up? How many of you here have been involved in scale up? How many has been involved in going from a small scale?
The starting point is, do we really do development?
Now I'm moving away from development.
I know, but in terms of what I'm trying to say is I don't know, I've left the industry for quite some time, the local arm. I was in Speaker before I moved to Novartis, and then I was actually the one that got the WHO to give them the technical support before I left from Novartis.
They should give you money, that's good. Okay, go ahead.
So what I'm trying to say is, in terms of the generics we have, do we really have a process of developing that formulation?
Yes.
And I mean, in the country, this is a question to all of us. I am learning, we are all learning. Because what she is trying to teach us is very important. If you don't go through this process, there's no how you'll be able to say, okay, the paracetamol we have in the country, are they interchangeable with Panadol, the innovator? So let me just put it like that.
That's
I need bioequivalence there. Yeah, bioequivalence, that's interchangeability is another WHO word for bioavailability too. If your product is not bioequivalent with the innovator, then your development process is flawed. Your formulation is flawed.
Thank you very much for bringing that up. That is where, before you try to copy the innovator, you have to understand the excipients. I mean, I started with pre-formulation, and then what I tell the formulation scientist that will finish it must be based on my data. if I do not give them the green light to use starch, for instance, and why there must be data to back up why I'm telling them that. So if you are able to study your excipients and then to avoid patent infringement, if you are able to avoid patent infringement, you have to use data to support it. And that's doing the excipient compatibility studies, drug excipient studies. But by doing that, I know most times people think it takes time, but it's something you can understand by, please, understand your process, understand the material. Because once the agencies are asking questions, they can tell that you don't understand what you actually put together. And if you are able to understand your processes and then understand your product, they will see. There's no argument. I'm happy that you were part of that. If you do not provide those information, you are wasting your time. And once they refuse to accept, you are done. You are go back to square one. So these are the, is it developable? How does it perform? Does it match the innovator? But this can only be answered if I did my prior studies to understand what I need, to understand the properties, and to understand the interaction. Okay, so you see here, just performance is how does your solubility go? Developability, how is it stable? And these two things have to work simultaneously. Must. Let's go. Okay, so this is the roadmap to your actual formulation. If you don't have your pre-formulation understood, everything we've been talking about is like, pre-formulation. So, what is pre-formulation? The strategies are the pre-formulation activities where you introduce everything. If you want to use sea salt, you want to use the other salt, but you have to know why you choose the sea salt. And then what's the biopharmaceutics, the BCS classification, and then the excipient strategy. There has to be formulation strategy and there has to be analytical strategy. And those two will now satisfy the regulatory strategy. But if you have problems in those two strategies, you now there has to be collaboration between the agency and the formulation so that they can know the challenges you're having and they can suggest. But like this man said, you lead them. You tell them what you believe is right. And if they know you understand, they will bow. They will follow you. But if they know that you are a monophobic, sorry that I'm using, I try to practice my Igbo language when I'm outside the US. If they know that you know your stuff and you have tried, I don't want just to put things together and say, let's go. If they know it's the agency like that, they will actually come to you. They will ask you to explain. And because they are going to use you as a benchmark, just like we are using SWIFA now as a benchmark for other organizations to be able to achieve WHO. If you're able to have all these things done, then of course you have a flawless formulation. So my message is do not avoid understanding your material, understanding the process, and understanding the product. So it says here the pre-formulation is the initial critical stage. If you miss pre-formulation, you are done. I mean, you are not going anywhere. So it says it's the initial stage in drug development where the physical, chemical, and mechanical properties of the API, a new candidate, are investigated and characterized. Yeah, the innovator has done it, but now when you are doing the generic, do your own due diligence to understand what they did. They call it reverse engineering. Even people that sew shoes in Aba, I understand they go to Italian shoes, they buy Italian shoes, they pieces it, and then reverse engineer and sell it to you in the market. So these are the things, API characterization, solid state properties. We talk about polymorphism and all that. Polymorphism, my advice for It's not that you should avoid any product that will create problem in terms of polymorphism. It's just the ability to have a product out there. You can look for the ones that are easy. You can say, look for ones that you can push out so that you are comparable, competitive elsewhere. If you get the ones that are very complicated, it's difficult. Then why do we do these excipient studies? It's to identify early risk. If you do not identify your risk at the beginning, at the end you have the problem. Every effort, your time, your money is zero. You've lost. And most times, some companies go bankrupt and they will lay off staff. Okay, let's go. Why are you doing the pre-formulation? Basically to avoid the failure risk at the end. That's all this thing is talking about. Yes. So these are the what must be understood and why does it matter. So everything I've been talking about is what must you understand, like the particle size, the flow properties, the polymorphism, and all those things. The excipient compatibility studies, physical, chemical properties. So these are, but why does it matter? If you take number 2, API excipient compatibility, it prevents avoidable formulation failures. That's why you have to understand it. And then likely degradation pathway when you do the studies. Where does Africa or Nigeria fall into the climatic region? The stability studies, is framed and set up, designed differently from, let's say, even some parts of South Africa, or let's say in the West, in UK that is not very far from here. So what I'm trying to say, why these things matter, each one of them, anything that I'm saying here, there is a reason for you to do that. So why it matters is, the studies will help you to choose the right dosage form. Because remember, you're thinking about the patient as a patient-centric opportunity. You want to understand since I've done all the studies, which one do I choose now? How many patients tolerate the injections, or what do they tolerate, the solid, the capsule, or whatever. And then, most importantly, preventing formulation failure. Okay, so we are now in the BCS classification. Let's go straight to the next slide. So here there are 4 classes. This is very important. If a product is the solubility, the permeability, the example of the drug, and the formulation approach. If you understand your BCS classification, then you'll be able to know which approach to use. This should be your Bible as well. You should understand this. Maybe in your wall somewhere, just put the BCS classification so you can tell. So if these two, can you tell me why Panadol is very easy to make, or paracetamol? Why? Come on, you should know this one. Yeah? Yes, you see, class 1, both of them are high. So it's easy. That's why, so you look for areas of least resistance. This one is also ibuprofen. There are challenges just because it's low solubility, but you have to do your characterization to look for something that can help you. That's where all the initial studies come in, because you understand. Then the class 3, if it is soluble but it's not available, it's not permeable, you have to also do a lot of tweaking. Tweaking means you can add some surfactants, you can add a lot of things, surfactants to help you to change it. So these are the things, the fundamental things you need to know. If it is low and low, there has to be some approach you use. It will work. But those two you can easily compete. Okay, this is a more detailed version of the other. this one gives you. Do you people get the slides later? Yes. Okay. So let's keep pushing. So role of excipients in formulation design. So there you have the binders, you have the disintegrants, you have the fillers. So all this, for instance, the binders, what are the primary function? So when you are doing your studies, you have to look at the properties of the API, and then figure out what type of binder you can use. So the primary function, the excipient behavior can help you determine which one that will, you will really want to pursue. And then we have a lot of raw materials in the country. So the Nigerian source potential, cassava and plantain starch, these are something that we can actually export into other countries. Well, we're not doing the right things. That you have modified corn or plantain starch, all these things, palm oil, they are, it's a very good, we have everything that we need, it's just to study it and understand it. And I hope that in the colleges, students will be able to do the basic stuff. So it says here, cassava starch, they have studied this so much in this country. In fact, every product we have in Nigeria is highly, even Chinese people are studying it. Okay, so now we are going to the core formulation principles. So this is a roadmap. So you see, first of all, we need to decide the drug we are going to make, what will be the route of administration or route of. How do you pronounce it? Route, right? Yeah. What's the route of administration? Will it be parenteral? Will it be oral? Will it be an inhalation? So these are the things. If you just have this pasted on your, maybe at your dashboard anywhere, anytime you want to propose a project, these are the things that will guide you in helping your organization to make that decision. So what type of release mechanism are you looking for? Immediate release or modified release? So if you have that, then you already have an idea. You remember it says first source, the targeted. And if you want to avoid the patent issue and all those things, yes, if they had immediate release, you can create modified release. If they had modified release, tweak your own. Look for something that will give you, so you will become competitive. And the development, there are 5-stage process map. We will look at it later, from formulation to scale. And then the analytical cycle, cycle, which we call analytics. So when you talk about analytics, you are now bringing the data science, you are bringing the artificial intelligence, which has actually blown up. Do not be afraid when we are talking about AI. You people are much younger than I am, and I have embraced it, and I'm running it. If you do not embrace it, then you will drop off by yourself, because you can't. And now in the world, it's the Indians that are teaching it. They are teaching it night and day. And I asked one of them, I said, how did you learn what you're teaching us now? I did a course with MIT, executive course. It took me 6 months. My head, all the hair, in fact, that was why I cut my hair. Because it was so stressful. But I remember that I went to pass. But most of my classmates, most of them dropped. Some of them sued the university. That it is very hard. But it's something that you must embrace. You people are very young, so it's like drinking Coke for you. And then, see, the QBD overview will help you kind of understand this thing, and it will make life very easy. We'll see how it works. If you understand the classification, you can, there's one page that you just press it somewhere. Even me, I still look at it if I'm trying to make a decision. Okay, let's push. So yeah, this is talking about Okay, now we come to the meat. For this meat, I will not go home, finish everything, but that's part of the thing we'll expand tomorrow. So, what is QBD? See, I highlighted these things so that if you can memorize this, quality by design, it is a systematic approach. Systematic approach to formulation development. And how does it start? By predefining your material, predefining your objective. You have to, before you go to say you are going to cook bitter leaf soup, what type? What are the ingredients? Do you want to cook the Imo State bitter leaf soup, or do you want to cook Anambra's? And depending on what you have, then you look for those materials. So, predefined objectives and emphasis, and emphasizes product and process. Product and process understanding. If you do not, that is, you are wasting your time. If you understand the process, then that will help you to have a control of your process. Like if you are doing scale up in the manufacturing, then or even everything has been processed, and now you have a design space, and you're now working on those, and then all of a sudden you see temperature start moving around, going high or dropping. It means your process has not been controlled. So you have to evaluate those processes and make sure you understand the process. And this is only based on science. Is it sound science and quality risk management, quality management. So this, once you have this in your mind, what is QBD? This guy that talked last, he didn't use the word QBD, but he basically set his own parameters. And that man was amazing. Okay. Do you have any question? Have you, in your work and in your job, have you used this before? That will help me figure out where we are. How many of you have actually embraced QBD?
Quality by design.
Yes. So what do you understand? Please share with us. Because once he knows that you know it, he will go home and say, that guy was able to answer. So I should be able to be like him. You know, you were competitive when you were children. I don't want anybody to. So can you tell us?
Yeah, it starts from when you want to design any product. You start from building all the facts and all the
from the start of your developmental process. You highlight all what you want, the functions, how you want the product to perform, what is the target that you are setting out, what are the parameters you want to check out for. That's in terms of the analytical procedures, and how do you want the product, the stability, the permeability, solubility. So when you are designing, you factor all this into it. So your design starts from, and the quality starts from the design. It's not after
listing everything.
Yes, everything is listed. So it starts from the quality of
that product. Starts from the design itself.
not after the product has been produced or formulated, that you are not testing quality into it.
Great. You know, that is the mantra. The old process is you test until you get the right result. But the real QBD process or approach is you build quality into the product. And that starts by selecting all the variables, all the materials, and evaluating those materials, understanding the materials, and know that it will meet the target or the performance that you want. Once this is created, and then you now build your product, and then the product, you test it. And I'm happy you mentioned the analytical. The analytical, we'll get to that slide soon. Please, all these ICH guidelines kind of contribute, QBD, make sure that you meet every requirement of this. It says ICH guideline is internationally harmonized science-based standard developed by the ICH. It defines technical requirements for pharmaceutical development, manufacturing. The Q series are this. There are Q series, I think it's 1 to 14 or 18, I can't remember. But all this, so, and it's also the quality control to ensure consistency, stability and safety. So to use, do you have a question? Okay. So all these things together, extracting each one of them will help you achieve QTPP. So you see, SCH guideline is talking about the QTPP. Who knows what is QTPP? Okay. The QTPP, the CQA is the critical quality attributes, the critical process parameters, the critical material attributes, the drug substance, and the control strategy. So the analytical has something similar, but they have different compared to that. So the quality risk management is being able to review and catch risk at the beginning. Once you open your eyes, make sure you are looking for any loophole. The pharmaceutical policy system is the life cycle approach. How am I able to start from, remember we're talking about end-to-end development here, or end-to-end, so that once you manufacture, you are able to have all the initial strategies, you know the materials, how you put them together, how you are able to produce, how you are now able to maintain post-launching. Once you are selling it in the market. Remember that when people take their medication, they have problems, they report it to the agency. And that's why sometimes they have drug recalls, right? They do drug recalls in Nigeria. Okay. So it's such things. And then the ICH, development of manufacturing of drug substance. And then the QB approach. Let's push. So what you must know, this is like the Bible. And why does it matter in Nigeria? The international, the regulatory bodies, which includes your NAFDAC, FDA, Japan, all of them have their own, but at least they have a lot of things in common. They require this QBD style submission, whether you like it or not. If you do not meet these ones, don't even try. If you try, you are just humiliating yourself. The QBD, and why it is important, is that QBD reduces the risk. If you didn't get anything else today, just know that QBD principle, QBD approach helps you to reduce risks. The risk that you will see, let's say in 9 months, you have seen it at the first month, and then you change, and then you are still within your time, and then it positions your product for ECOWAS and global export. That's the WHO framework. If you are able to do these things, you know you are good. So I put here that full dedicated presentation will be there. Let's go. So here I put that the drug formulation, there are two things that are happening at the center. Even if it's just your drug, your excipients, you still have to test your excipient. You have to test the material. You have not mixed it with anything. You have not mixed it with the drug. So there, from the get-go, you must have a tool to analyze your material. So whether it's HPLC, whether it's the simplest test you can do. Remember, there are some you have to look at it just by odor, sight. All those things are simple tests, but the analytics has to be there. So the formulation development, there are lots of activities that is going on, and the analytics is going on in parallel. So formulation development cannot be a standalone. Please hear this very clearly. Remember, quality is not tested in at the end. It is designed into it from day one. And so, if you don't do the formulation development and you did not test it out to know the materials, if it's good, and keep going, at the end you have made mistakes. So let's push it. So the QBD principle is that all these attributes, these are the things that you are looking out for. So the two of them go, it's a dual lane, I call it a dual lane process, lane 1 and lane 2. Lane 1 is your drug formulation and lane 2 is a analytics. And the pre-formulation there, so you can see the two of them are going on at the same time. The core science, scientific principle, the BCS classification, these are all the strategies. You know, the strategy, you need to understand this, your excipient, your selection, your formulation design strategy, the QBD, all those things you have to understand. And then the analytical strategy, depending on the type of product you have, then it will determine the analytical strategy you will use. And that will determine the type of data that will help you make a decision. And that's why I think the AI and hammering on data integrity or data science tells you from the get-go, do you understand your data? Do you understand it? Whatever you are talking about is your data that helps you to make a decision. If you have to go straight, move faster, your data will tell you go ahead. If your data doesn't tell you, you are not going anywhere. Okay, let's push. So this is the lane 1. Lane 1 has your pre-formulation, formulation design, process optimization, stability testing, and scale-up. And lane 2 is your analytical method. Your method development, which also you use your excipients to, if it's simple HPLC, to see how, do you get flat lines? There's no nuggets you have. Lane 1, these are the processes. Lane 2, these are the processes. And they marry each other to be able to. Okay, so let's go to lane 2. Okay. So every measurement must be accurate and legally defensible. Why did I say that? And I've gone to represent my company in legal, when they sue you to court. The first time I tell you I was the expert to defend, I almost peed on myself. I'm just being very blunt. I forgot the address of my home because the attorney is trying to just mess your brain up. They asked me, "Where can you give us your address?" I forgot my address. And so there are two attorneys from the company that's with me. They told me when I feel stressed, I should just tap the tap, so they know to say time out. So they take me out to kind of reduce the sweat. So legally you have to defend anything, any data you generate, even with your boss. I mean, there's a product that I handled, acetaminophen. All of you know acetaminophen, right?
Yes.
You know that there's an impurity, impurity in it, right?
Yes.
I won't say anything that will scare you guys, but it is the most used API in the world. And I used to work in a company called Malincott. And Malincott distributes it all over the world. So when I joined, they actually hired me from Johnson & Johnson because of another product. But this particular acetaminophen, they were selling it to buyers, other companies, and giving them certificate of analysis with wrong data in it, wrong results. They were producing 25 parts per million of the paraminophenol. And so when they were, FDA said that they want everything to be below 1 part per million. So they pulled me into the project because the man that was handling for 2 years, they couldn't go anywhere. So within 1 week, I was able to come up. They are actually initiating the breakdown of the acetaminophen. Acetaminophen. Yeah. So it was breaking down. So I saw that they were doing something kind of crazy. They were using methanol and a lot of things. So once I changed that, we were able to go to 0.6 from 25. And so I went to present it to management. They said, Nonye, you are turning our company their head upside down. What do you want us to tell all these people who've been selling these things, giving them, see your face? I said, that's maths, go and so they brought 3 chemists from the lab and asked them to repeat the test. They were getting 0.5, 0.6. I basically explained to them, this is what you have been doing. But that thing cost, I got some money out of it anyway. They gave me $15,000. So, because I basically saved them, they were poisoning, but they don't call it poisoning, but they were creating problems. So every data that you generate, you have to defend. And as a black woman, in fact, that was when I went to the attorney, that was the first time they were calling me Dr. Onyewuenyi, Dr. Onyewuenyi. What Dr. Onyewuenyi said. But before that they just called me Nonye. But once they see that you are talking, then they know, they will call you and they will do everything. But it stressed me out. Why am I saying this? Every data you generate, you better back it up. Make sure it's accurate. If not, they can fire you or you can be an expert witness. Okay, so QBD has 2 domains. Domain 1 is the main 1 and domain 2 is the analytical QBD. They use the same principles except that different things. You have to start from the beginning. Look for everything. Then if you go to analytical, let's say analytical method. For instance, let's say the simple HPLC. You have to know every type of, is it the methanol, what type of methanol, all those things. Water, even water, there are different types of water. There are so many. So those simple things you need to understand and evaluate them. And then when you talk about the system control, in HPLC, before you shoot your first injection, remember you have to do system suitability. If the 6 gives you less than 2%, right? Then you know that you are ready. The same thing with this, the process analytical technology. If you are able to have your design space, everything is working. If it is not working, you have to recalibrate. Just like in your HPLC, if it is your system suit is giving you 2.5, know that your system is bad. Either there's a leakage, there's a lack, the detector is not good or something. I'm running fast so that okay. So the application of plants is looking at the risk, like the QBD, the parts, and all these things, they are all applicable. How do you achieve products and process understanding? So these are all these things. I'm going to shout their hand off so that they can.
No, give me something under the end.
Okay, you see, for formulation, the design space is called method operable design region. So as you're working on your formulation, make sure your analytical department, they are supporting everything you're doing, because if you do not have robust method, it may not be fully validated, but before you go and do your final whatever and submission, there must be validation. But it helps you to make sure that you're on the right path. This too is very important. I think we'll see more of that tomorrow. Okay, let's see. The Six Sigma principle, I actually did. Most companies in manufacturing is very important. What goes to manufacturing? Actually, there were 4 of us that were sent. It's very difficult to do some of these things. But as a black woman, when you're in a foreign country, you try your best not to fail, so that you get money. Because that's the only way we survive. So I had to do this, and then as soon as I came, there were 8 departments of formulation scientists that were doing different products. So they made me to work with each one of them to know if they understand what they're doing. After that, 2 people quit by themselves. Because you need to understand the process. It's almost the same thing we are talking about. You need to understand the process. But this one is I use something DMAC. Define, measure. This one was talking about measurements. Measure, analyze, improve, and control. This, even if you're sleeping, should be able to understand these basics too. And then the same thing here. Define your target. Your QTPP, you must understand the quality target product profile. You want to determine the critical quality attributes of the in-process and the finished product. And then you also have to understand, do a risk analysis to make sure that your materials, number one, the source, and number two, it doesn't have, maybe it's not too hygroscopic when you need it to be maintain, dryness. So all this, then this one, I know that many people are not. I'll try to explain a bit more tomorrow. The DOE, how many of you have used the design of experiments before? Yeah, it's very important. Before, even now, some people use one factor at a time. Let's say you want to know how many meals of, can you give me an example? Let's say I want to use 1 gram of salt in my Facebook page. So I'll do that 1 gram and run the experiments. Maybe it takes me the single factor. Yes, single factor. Or as we call it a fact. One factor at a time. You waste time for 2 weeks, 1 month. But using the design of experiment, you can do 2 or 3 factorial design where you have 3 factors or 4 factors, and then all of them, you look at the interaction between the factors, and then you can pick. You see it's very easy. So these are things that you have to do. Then once you are able to do it, how can you implement those controls? The same thing with this. When you control it, then it means that your future state is guaranteed. Because I'm already sure what I have done. I've evaluated everything. So let's go to the quizzes. Okay, that last one is just problem solving tool. Hopefully, how many of you know about agile methodology? Okay, Scrum Master. No, so I see you nodding your head.
Yes.
Okay, we'll go back to it. So let's do a quiz and see where you are. So a drug with high solubility and high permeability belongs to which BCS class? Okay. Okay, so the way I did this, so did you know that BCS class 1 drugs like metoprolol and paracetamol are the dream candidates for oral formulation? Just because of those 2. That's why I said it's low-hanging fruit. If you just find something, because once you start, you can sell your products overseas. All these African countries, there are 52 countries in Africa. Once you can produce your own paracetamol and push it out, you will be swimming in millions. And that is, you can only do that if you are WHO qualified and they give you that. But they make you sweat, just like I'm sweating. Wow. They make you sweat. Okay, so which locally available Nigerian agricultural material has been the most extensively studied as a pharmaceutical binder? Yeah, everybody's, yes, that's good. Okay. So cassava starch from Nigeria has been extensively studied. And this can be marketed, pushed out, depending if you're able to kind of support and do all the needful. Let's go. Nigeria falls under, I asked this question, what climatic zone? The most demanding stability testing category in the world, requiring testing at 30 to 75 relative humidity. True or false? Okay. Didn't say anything. What about your friend? Okay. So true, Nigeria's hot and very humid. Hot and, when I mean humid, places it firmly in zone 4B. So any stability studies you have to plan will have to be, taking this into consideration. Let's see where we are. Maybe I'll go straight to, how many minutes? I'll go straight to, I'll fill up Okay, so that's the first. Let's go to, yes, this one. We'll finish it, but maybe tomorrow we'll do a lot of case studies because they are all related to both of them. So, innovation and credible laboratory. I intentionally put the credible laboratory here. The credible laboratory, if you cannot test your data and it has data integrity, then you don't have a problem. You cannot say you are being innovative. So you want to, is one of the things I'm proposing, that we must have a credible standard, credible laboratory that we know that they measure everything. It assures us everything is what is supposed to be. And if you have a credible laboratory, it will help us to have AI-enabled facility or processes. Let's go. So from innovation to implementation, there has to be, because this conference is talking about collaboration and innovation, there has to be a credible laboratory to be able to. So pharmaceutical innovation defined as meeting unmet medical needs through efficacy and those other things. Novelty alone does not qualify. That is new doesn't mean anything. It requires novelty of effectiveness. Okay? So what makes a credible, a laboratory credible is, who knows about the ALCOA principle? ALCOA principle. That's why I'm saying actually. Yeah, you feel like saying they manage people. Yeah? Okay, yeah. So accuracy and precision, the results approach the true value. Repeated analysis must give you a consistent result. And then sensitivity and specificity correctly identifies the individual peak you're looking for, whatever, consistently. Not today, or Mr. A does it, or Analyst A does it, Analyst B. They have two different results. And probably they use the same mobile phone, the same everything. And you know it's not a credible lab. And then GLP and accreditation. So recognize national and international accreditation for each test method. Your credible laboratory can only ensure that. And so data integrity, you talk about attributable. Do you know what attributable means? Whoever does the test, you can attribute it to that person. If he did the test and he made mistakes, they can't come and catch me. He's the one that did it. So anybody will, and then legible, you have to be done scrabble like a chicken. It has to be legible, not just typing. If you write, some people are good, I don't write that. It has to be contemporaneous, original, accurate, complete, consistent, enduring, and available. Anytime an auditor wants to see your data, there's no running around looking to hide the result. It has to be produced. So Nigeria's path to innovation and everything they want to achieve, they must build credible laboratory which ensures data integrity. And this can be digitized, and this will be leveraged by artificial intelligence and machine learning. And I think that's the next thing. Okay. So please, I urge you to have an open mind. And you see there's a lot of advantages to this. AI and machine learning in drug manufacturing. There is a big paradigm shift. And you see, when you catch the bug, you see that all the things we have been doing, we have been struggling for nothing. But once we are able to use the right tools, it works like magic. Somebody was saying something yesterday. The woman, I can't describe her. I can't describe her. Okay, let's. So what is AI ML in pharmaceutical manufacturing? That paradigm shift is shift from trial and error to data-driven. You can be able to, we'll see what is prediction when we go to one of the case studies. Predictive drug design and manufacturing. So no more trial and error. We want to use data and make our decisions and move. And then artificial intelligence is computer system that simulate human intelligence. In the Western world, a lot of agentic AI, they create these models that behave like you and I. And it's going to get worse. But don't be afraid. You'll survive. So it reasons, it learns, it recognizes patterns and all that. But the most important thing is that it helps us in drug manufacturing. The machine learning, on the other hand, is a subset of the artificial intelligence, where the algorithms learn patterns. You train it, the machine learning, you train that model until with a set of data. You can, yesterday they were talking about they can file the CTD or package the ECTD quickly within seconds. It's because these models have been trained. You can actually dump a whole chapter and it will synthesize it in 1 or 2 sentences and pick out the errors that are there. And so then the deep learning is a subset of that. Let's go. So I want to get this one. So why artificial intelligence? Why? It's a compelling case for pharmaceutical manufacturing. Maybe in your different organizations, you can be, if you pick up a few things here, and maybe in the future there could be more training, I don't know, you will be the proponent in the organization. And just, they look at the money to develop one product. It's costing $2.6 billion. This is in dollars, not in naira. That's why when brands release their products, they want to recover the money because it takes from 5 to 17 years to do one product. So working in brand takes a long time. So these are all the things that's driving you. So now, if you use the AI and to address those challenges, then you can see there's, for instance, people in drug substance. The drug substance is very, it helps you to make decisions. In fact, during one of my training with the MIT, I had to propose develop certain groups of models that can be used by pharmaceutical companies to identify a contract manufacturing organization that has zero defects, zero problems. Whereas without the machine learning, you will go and try this particular, let's say, contract manufacturing organization one. After the first 3 months, we found out that all their development, they messed it up. But with machine learning, there are some factors that have been created, which is what I can credit to my name. So it helps you to predict the future, predict whatever. So this is very important. So let's go because I want to So this is how it works. Data ingestion. It can take all your lab results. It will process all the data and be able to separate which. The good thing about the AI is that it can process your data to a clean and easily interpretable data that can be used for decision. And then it helps you in model training. But most importantly, whatever you are using has to be deployed. Deployment is the last. If you don't deploy your process, you have not done anything. There has to be deployment. So the core techniques here these are the different ones. Don't worry about this. I just wanted to bring it in so that, because they're talking about innovation. Innovation, it's already started. Without AI, I don't think we'll move anywhere. How it works. It trains on label data. And once they are trained, it's easy to do things something that can take you 3 months to do, it can take you less than a day to do because it has been trained. So in pharmaceutical, how do you apply that? Formulation failure prediction. If you are going to have a problem with your formulation, the AI will help you to predict it way early. and so it tells you what to do. For instance, in your dissolution method. And then the molecular property can help you to predict it. And then it helps, it knows how to mine the literature, the bucket load of chapters and all that. AI will replace pharmacists and formulation scientists in Nigeria within 10 years. Is it a myth or a fact? Okay, that's very good. Okay, so that's good. Let's move. And then Nigerian pharmaceutical manufacturers are too small to benefit from that. Is it a myth? Okay, and the last one. AI has already been used to help develop a pharmaceutical product approved by major. Yes or no? Or major? It's a fact. So it's being used and it does it very quickly. This one has to be, it has not been established, but I know it's there in different bits and pieces. So the credible laboratory will use data science, data integrity, we'll talk about data security. You know there's a difference between data security, data quality, data safety, and data management. So this is very key to whatever. Can I see the next slide, please? Let's go. Okay, this one you can see the data. Formulation excellence is not merely a technical function. It is a strategic capability that underpins everything. If you are able to understand your formulation development, most of the time now you're just doing manufacturing based on the innovator. But if you are able to do it, then you have a self-reliance approach. Let's see. So the key takeaways here. Pre-formulation data helps you to make formulation decisions. The dosage form and the route selection must account for Nigeria's patient population. Must. And Zone 4B is critical in designing your stability studies. How many of you have done worked on stability studies before? Okay. If it's not designed properly, the data you generate is useless. So artificial intelligence and machine learning represents a major shift. It's a major shift. So Nigerian manufacturers who adopt now, any Nigerian manufacturer that adopts now will own a decisive competitive advantage. Mark my word. They will own a decisive competitive advantage. Even going to WHO pre-qualification will be a breeze if you do the right thing from the get-go. So local excipients from Nigeria's rich agricultural base can significantly reduce import dependency. Everything you could import from India, crazy stuff. And then collaboration. I don't know how many, most companies do not collaborate. There are some people, some managers that do not want to collaborate. Even within the company, some people are just, they want to so collaboration between the industry, and international partners is the single fastest accelerator. Single fastest. So if in your own little world in your company, you can make proposals. Remember what is called ideation, right? Ideation. Always have a notebook. I'm giving you my secrets. I schooled in Australia where I was the only, always anywhere I am, I'm the only black person. And I moved to US. Every company, I'm the only black person. I'm the only one that has a place, a woman. So I'm telling you this how I managed to survive, even at my old age. So you have a notebook, anything that comes to your mind, note it down. And from there you can build it and present it to your management. And you will say, if you don't remember anything, remember what this woman is telling you. Ability to ideate. Ability to ideate is very critical. Especially in this day and age of artificial intelligence. And do not be afraid. The Indians don't know more than you do. I battle with them every weekend, Saturday and Sunday. So sometimes they say, Dr. Onyewuenyi, you're always asking questions because I want to ask. If I don't ask, nobody will tell me. And so when I ask, they hide behind me to get the truth that I squeezed out. So trust yourself, and we'll be able to. When we come tomorrow, because QB is part of this, we'll visit some of the case studies that we will cover today. So make sure, now that we have adjusted to the environment, adjusted to the disruption, because this is a real disruption, this environment is a real disruption. We'll start much earlier tomorrow, and we'll make sure we'll finish what I prepared for us. Thank you. Thank you very much.
Dr. Nonye Onyewuenyi, please put that together for her. She started by saying that when you can produce a good product, nothing stops you from marketing it in wherever else you want to. She has given us several steps, and that pre-formulation is a prerequisite to your formulation because they can determine errors early and reduce costs on the drug discovery. She also talked about quality by design, where you're building quality from the beginning and building also analytics from the beginning. That can also reduce your costs and reduce failure. And finally, she told us that AI has come to stay, that we as pharmacists, we should not be scared and we should not be shy to embrace AI. AI is here to make our life better, to make our work simpler. And they give us faster discovery, they can predict failure, and they give us process optimization and knowledge mining. AI can scan several thousands of applications and generate information that can use in drug discovery. Thank you so much for listening. And let us, if you have questions, I think only two questions, please.
Yeah, thank you for the wonderful presentation. Please, I don't know, sorry, there's this 3D in drug design and modification. Please, AI and NL, are they facing out the 3D, or 3D is part of the AI? I mean, 3D, is it the matrix of the AI, or 3D is different from AI?
The machine learning or the deep learning is a subset of the machine learning. So the 3D you are talking about is just the presentation of, when you are kind of designing the model, whether the model will be a 3D version or mode. Does that make sense? It's not something that has a major distinction, but if you understand the ML, which I don't know if you're doing it already, but no, you are still at the infancy stage, but it's good to do those things. The deep learning is just a separate version. There are so many things within the machine learning.
Thank you, Doc, for the wonderful lecture and presentation. Really appreciate. So my question actually is, I would say the introduction of AI into manufacturing industry is a welcome development in the sense that it helps us to mitigate some loopholes, avoid errors, and production processes are becoming more efficient. But my concern is, there is an info I have, even in the US, as of the year 2025, November 2025, unemployment rose from just within within the year 2025 in the US, between 54,000 to 141,000 people became jobless actually. So introduction of AI into their industry. So yeah, my question is, are there measures being taken probably by the government, or are there things to be done to help mitigate the issues of unemployment?
Thank you. I can make a little contribution. Someone asked me that question sometime. Will AI replace professionals? AI only replace professionals that refuse to upgrade. AI has come to stay. If you don't upgrade, it will replace your job. So when you upgrade with AI, your job remains with you and you get relevant to the system. That is my little contribution. Anything else?
I just add that, like I said at the beginning, do not be afraid. But it is come to stay. In terms of will it replace human beings? Ideally not, because there's something I say, there's human in the loop. AI will still make some mistakes, and if you read some things, AI will generate something I will tell you. Read between the lines. So the only thing I would advise you, you have to upgrade. You have to study, be open. Yes, even the employers they don't really know what to do. Like there are classes I handle Saturdays and Sundays. The management companies, they don't really know. They are spending money to send their employees to learn AI, but they don't really know the outcome. But it's something that every day something new will unfold. But do not stand and watch.